Good afternoon. This time we will discuss recombinant DNA. Recombinant DNA or rDNA molecule are DNA molecule formed by laboratory method of genetic recombination that bring together genetic material from multiple sources, creating sequences that would not otherwise be found in the genome. Recombinant DNA technology comprises altering genetic material outside an organism to obtain enhanced and desired characteristic in living organism or as their product. This technology involves the insertion of DNA fragment from a variety of sources, having a desirable gene sequence via a appropriate vector. A DNA fragment are usually derived from different biological sources. This table is represent the brief history of recombinant DNA. The first breakthrough of recombinant DNA technology occurred with the discovery of restriction endonuclease or restriction enzyme during the late 1960 by Warner, Arber, and Hamilton Smith. The restriction enzyme were discovered in microorganisms. This enzyme protect the host cell from the bacteriophage. The restriction enzyme are described in the preceding section. Then in 1969, Herbert Boyer isolated restriction enzyme EcoR1 from E. coli that cleave the DNA between G and A in the base sequence G, E, E, T, T, C. In 1970, Howard Temin and Devin Baltimore independently discovered the enzyme reverse transcriptase from retroviruses. Later on, this enzyme was used to construct a DNA called complementary DNA or cDNA from any mRNA. In 1972, David Jackson, Robert Simon, and Paul Berg successfully generate a DNA molecule. They follow the sticky-in of complementary DNA by using an enzyme DNA ligase. In 1973, for the first time, as Cohen and M. Boyer developed a recombinant plasmid. The name of this plasmid is PSC101, which after using a spector replicate well within a bacterial host. And in 1975, Edwin M. Soughton developed a method for detection of specific DNA fragment for isolation of a gene from complex mixture of DNA. This method is known as the Soughton blotting technique. Then in 1982, commercial production of coli of genetically engineered human insulin, isolation, cloning, and characterization of human cancer. The principle of recombinant DNA technology involves four steps. The first one is gene cloning and development of recombinant DNA. The foreign DNA, in this case a gene of interest, from a source is enzymatically clipped and ligated, in this case, joined to another DNA molecule. For example, a cloning vector is a plasmid, gemide, etc. to form recombinant DNA. Then the second one, transfer a vector into the host. This cloning vector with recombinant DNA is transferred into and maintained within a host cell. The introduction of rDNA into a bacterial host cell is called a transformation. Then the third one is selection of transfer cell. Those host cells that take up the recombinant DNA are identified and selected from the pool. And the last is transcription and translation of inserted gene. If required, and recombinant DNA construct can be prepared to ensure that protein product that is encoded by the clone DNA sequence is produced by the host cell. Transcription of DNA into mRNA is mediated through the enzyme RNA polymerase, which recognizes the binding site of on DNA called a promoter. The process of mRNA synthesis is terminated by a termination signal or a terminator of codon. This means only gene lying between promoter and a terminator will be transcribed. Gene isolated in a certain way, such as cDNA cloning or artificial synthesis, do not have their own promoter. Therefore, they must be inserted into a vector close to a promoter or site. There are three different methods by which a recombinant DNA is made, such as transformation. The first step is a transformation is to select a piece of DNA to be inserted into a vector. The second step is to cut the piece of DNA with a restriction enzyme and then ligate the DNA, insert into the vector with DNA ligase. The insert contains a selectable marker which allows for identification of recombinant molecule. The vector is inserted into a host cell in a process called transformation. Then Bagi introduction. Bagi introduction is the process of transfection, which is equivalent to transformation, except a bagi is used instead of bacteria. This process uses lambda or Mi3 bagi to produce bagi plug which contain recombinant. The recombinant that are created can be identified by difference in the recombinant and non-recombinant using various selection methods. Then the last is non-bacterial transformation. By using micro-injection, the DNA is injected directly into the nucleus of the cell being transformed. The host cells are bombarded with the high velocity micro projectile, such as particles of gold or tungsten that have been coated with DNA. There are several steps in recombinant DNA. The first one, isolating. This is a vector and a target gene. Cutting, clipping, amplification using a PCR, joining or ligation, insertion of recombinant DNA into host, and isolation of recombinant cells. Now let's see isolation of DNA. The first step in recombinant DNA technology is to isolate the desired DNA in its pure form. Since DNA exists within the cell membrane along with other macromolecules such as RNA, protein, and lipid, it must be separated and purified. It must be separated and purified with involved enzymes such as lysozyme, cellulase, chitinase, ribonulase, and protease, etc. Other molecule, the other macromolecule are removable with other enzyme or treatment. Ultimately, the addition of ethanol causes the DNA to precipitate out as fine thread. This is then spooled out to give purified DNA. Then cutting or clipping. Restriction enzyme act as molecular scissors that cut DNA at specific location. This this reaction are called restriction enzyme digestion. They involve the incubation of the purified DNA with the selected restriction enzyme at condition optimal for the specific enzyme. The technique agarose cell electrophoresis reveal the progress of the restriction enzyme digestion. This technique involves running out the DNA on an agarose gel. On the application of current, the negatively charged DNA travels to the positive electrode and is separated out based on a site. This allows separating and cutting out the digested DNA fragment. The vector DNA is also processed using the same procedure. Then amplification using PCR. A polymerase chain reaction is a method of making multiple copies of a DNA sequence using the enzyme. It helps to amplify a single copy or a few copies of DNA into thousands to millions of copies. The cut fragment of DNA can be amplified using PCR, then ligate with the cut vector. PCR reactions are run on a thermal cycle using the following component. First, a template. This one is DNA to be amplified. Primer, a small chemically synthesized oligonucleotide. They are complementary to a region of the DNA. Enzyme, DNA polymerase, a nucleotide needed to extend the primer by the enzyme. Now let's move to ligation of a DNA molecule. The purified DNA and the vector of interest are cut with the same restriction enzyme. The process of joining these two pieces together using the enzyme called ligation. Ligation involves joining up the end of DNA with other end. However, its DNA fragment has two in, and if the in are compatible, a DNA molecule can circularize by joining its own in. So in this case, the purified DNA and the factor of interest are cut with the same restriction enzyme, and this gives us the cut fragment of DNA and the cut factor that is now open. Hence, this new hybrid DNA molecule is also called a recombinant DNA molecule, and the technology is referred to as the recombinant DNA technology. Then, insertion of recombinant DNA into host. In this step, the recombinant DNA is introduced into a recipient host cell, mostly a bacterial cell. This process is transformation. Bacterial cells do not accept foreign DNA easily. Therefore, they are treated to make them competent to accept a new DNA. The process used may be thermal shock, a calcium ion treatment, electroporation, etc. So now let's see one more time this insertion of a recombinant DNA into host. See this one, host plasmid. This one, cut, then we'll get recombinant plasmid DNA. Now let's move to insertion of recombinant cell. The transformation process generates a mixed population of transformed and non-transformed host cell. The selection process involves filtering the transformed host cell only. For isolation of recombinant cell from non-recombinant cell, marker gene of plasmid vector is employed. For example, PBR322 plasmid vector contain different marker gene, such as ampicillin-resistant gene and tetracycline-resistant gene. When PS1RA is used, it knock out ampicillin-resistant gene from the plasmid, so the recombinant cell become sensitive to ampicillin. Now let's see the application of recombinant DNA. The first one, detecting the presence of HIV in a person. Second, in agriculture, for manufacture of a bit cotton to protect the plant against bollworm. In medicine, insulin production by DNA recombinant technology. Gene therapy, it is used as an attempt to current the gene defect which give rise to hereditary disease. And in clinical diagnosis, ELISA is example where the application of recombinant DNA. Then now let's see a limitation of recombinant DNA technology. The first one is destruction of native species in the environment. The genetically modified species are introduced in. Second, the silent plant can theoretically give rise to resilient wheat, which can be difficult to control. And the third, cross-contamination and migration of proprietary DNA between organisms. Then, recombinant organism contaminating the natural environment. Fifth is the recombinant organism are a population of clone vulnerable in exact same way. A single disease or pest can wipe out the entire population quickly. Then such a system might lead to people having their genetic information stored and used without permission. And the last, many people worry about the safety of modifying food and medicine using recombinant DNA technology. I think that's all for this afternoon. Thank you so much for your attention.